Drawing on his own observations as well as the clinical notes of both his father and grandfather, the original manuscript has annotations penciled in by his father, the young physician's paper offered a masterful description of what became known as Huntington's chorea, and now called Huntington disease. The chorea movements, he explained, gradually increase when muscles, hitherto unaffected, take on the spasmodic action until every muscle in the body becomes affected. He noted the attendant mental deterioration. As the disease progresses, the mind becomes more or less impaired, in many amounting to insanity, while in others mind and body gradually fail until death relieves them of their suffering. And he recognized that the disorder was inherited. When either or both of the parents have shown manifestations of the disease, one or more of the offspring invariably suffer from the condition. It never skips a generation to again manifest itself in another. Once having yielded its claims, it never regains them. Huntington correctly identified the key features of this kind of genetic disorder. He recognized that it affected both males and females, and understood that it passed from generation to generation. Each child of a parent with Huntington disease has a 50-50 chance of inheriting it. By the luck of the draw in some families, everyone is affected. In others, none are. If a person does not inherit the abnormal gene from a parent, he or she cannot pass on the gene to the next generation. Today we know Huntington disease is caused by a mutation, and since the gene is not preferentially expressed in one sex over the other, i.e. is not sex-linked, we have inferred that the affected gene is on neither the X nor Y sex chromosome. Let's call the normal version of the gene H and the mutant version small h. We have two copies of each non-sex chromosome, called autosomes, and so two copies of the Huntington gene. Individuals with the two copies of the normal gene, capital HH, are predictably disease-free. But individuals with two, small hh, or even one copy of the mutated gene, capital H, small h, are bound to develop the disease. We call this pattern autosomal dominant inheritance. Dominant means that only one copy of a mutated gene is sufficient to cause disease. The abnormal gene dominates its normal partner. Since it is far likelier that a person will acquire one rather than two copies of the mutant form, most Huntington sufferers are capital H, small h. Such individuals could pass on capital H or small h to their children, yielding a 50% chance that a particular child would be affected, just as Milton Wexler told Alice and Nancy. Back in 1968, not much was known about Huntington disease beyond these facts. It is heritable, and it makes its irreversible progress by killing nerve cells in specific areas of the brain. Milton Wexler resolved that he would take on the terror striking his family. He established the Hereditary Disease Foundation, HDF, to raise money and press for more government funding for Huntington disease research. His daughter Nancy was drawn in as well. While completing a doctorate in psychology at the University of Michigan, her thesis fittingly concerned the psychology of being at risk, she found herself increasingly involved in the affairs of the foundation. In the 1970s, when it became apparent that real progress would depend upon a better understanding of the genetics of the disease, Nancy Wexler began to reinvent herself as a geneticist. On the shores of Lake Maracaibo, Venezuela, the burden of grinding poverty is compounded by a remarkably high incidence of Huntington disease. If Huntington were to divulge its genetic secrets anywhere, Lake Maracaibo seemed a likely place. In 1979, Wexler began to collect DNA samples and to record family histories with the goal of preparing a genealogy of all affected people. For the geneticist, it was a great labor. But for Wexler, the daughter of a Huntington victim, with the possibility of the disease in her own future, it was more than that. It involved seeing the familiar in such unfamiliar surroundings. People who lived in tin-roofed wooden huts on poles above the waters of the lake, yet walked with that same drunken stagger that had overtaken her mother. Since her first trip to Lake Maracaibo in 1979, Wexler has returned annually to continue the work there. The people she works with have come to call her La Catira for her long blonde hair. As Americo Negret, her Venezuelan colleague and the scientist who first reported the occurrence of Huntington at Lake Maracaibo, describes it, she has made of them an extended family, greeting them each time, without theater, without simulation, without pose, with a tenderness that jumps from her eyes. But tenderness could only mitigate the devastation Huntington disease had visited upon so many. The goal of Wexler's expeditions was ultimately to find the gene responsible for the disease. But how could her Maracaibo genealogies help to identify the culprit? The key lay in advances in human genetics. If they were to home in on the Huntington gene, Wexler and others interested in genetic disease knew they would have to do for humans what Morgan and his students had started doing for fruit flies more than a half a century earlier. As we have seen in Chapter 1, Morgan compared rates at which particular genetic markers, white as opposed to red, eye color, say, and curly as opposed to straight wings, coincided in the offspring of crosses between parents showing various combinations of these traits. From these data, he was able to determine how near each other on a chromosome were the genes governing those traits. But human genetics had lagged behind the fruit flies for two major reasons. First was the impossibility, on moral and practical grounds, of doing the kind of experiments that were still the mainstay of genetic analysis. You can't simply breed two human beings you're interested in and then analyze the progeny 2 weeks later. Second, even if humans could be crossed at will, they were still lacking in genetic markers. Morgan was able to track a number of simple and obvious differences in appearance caused by specific mutations in individual genes. Humans, unfortunately, don't possess many easily analyzed traits that are inherited in this simple way. Even the canonical example, eye color, turns out to be governed by several genes, not just one. Furthermore, with fruit flies, you can increase levels of genetic variation by subjecting individuals to X-rays or to other mutagenic agents. Such options, happily, are not available in dealing with humans. Only with the advent of recombinant DNA did solutions of the two major obstacles present themselves. In the age of DNA sequencing, genetic markers need no longer be visible, like white eyes in a fruit fly. A variation in the sequence itself will suffice, and you can track such a DNA marker through a family tree. That is, through a number of genetic crosses, simply by analyzing DNA from several generations. The revolution had begun the year before Wexler started her genealogical research. And as with so many advances in science, a measure of serendipity